reproductive age. So that's overproduction of offspring. And you've seen this. If you've seen like cats or dogs have litters, they'll have 5, 6 puppies or kittens. And if they're doing that I think they breed once a year, maybe twice a year. And if they were able to have all their babies survive, we would be overrun with cats and dogs. So they produce more offspring than can survive. These offspring are not genetically identical to their parents or their siblings. So there's variation. And so there's different combinations of traits, and some of these may be able to survive better. Some of them may not be adapted to the environment and may die off. There is also competition. Since there are too many organisms to survive to adulthood, they have to compete for a limited set of resources, which means the ones who are better at that competition will live, and the ones who don't win that competition will die. And that all results in differential reproductive success. Not everybody has the same success at passing on their traits and their genes. So the ones who are best adapted to the environment survive. Not only do they survive, they reproduce, and they pass on their genes to the most organisms. Ultimately, if you think of evolution or natural selection like a game, the way you win is in 2 or 3 generations, more people have your genes than other people's. So if we're talking about evolutionary fitness, a person who has like 12 children may be more fit than someone like me who has 0 children. I know I have low fitness, evolutionarily and physically. But let's talk about an adaptation. An adaptation is some sort of trait that develops and is a favorable trait, as determined by the interactions of natural selection in the environment and genetic variation, etc. So an adaptation is a good thing. It helps you survive. There's also the fact that we impact variation in other species. So, for example, as predators, we have affected some of the populations in dramatic ways. So I'm not 100% sure about the status of this, but I know back when I was in elementary school and middle school, we learned that about 10,000 years ago, humans came to North America through the land bridge from Alaska, and very soon after that, all of the large mammals in the area died. So there were apparently, I think American cheetahs, there were American versions of horses, there were other animals like mastodons and the saber-toothed tigers and all of those, and they just all went extinct around then. So one of the prevailing hypotheses was we hunted them to extinction. And that led to a selection for smaller prey that could reproduce quickly and hide from us. We have something going on right now with fishing. So if you look at the news, there's all sorts of stuff about fish stocks collapsing and not enough fish for people to catch because we were pulling giant nets of fish from the ocean. And the ones we would keep, for the most part, were the big fish. We wanted the big fish because the big fish had more meat per fish, right? Well, this led to selective pressure for not only a smaller size, but also reproducing at a smaller size. So instead of waiting for sexual maturity until you reach this size, the organisms that would do that ended up getting caught before they could reproduce. The ones who reproduced earlier in their lifespan had a lower chance of being caught, so they reproduced and passed on their genes for early reproduction or small size. And so you've actually seen a change in the populations of fish just since the 60s. They've started getting smaller. We've also initiated the development of resistance in a lot of species. The big example of this is antibiotic resistance because it's a looming health crisis that we'll have to deal with soon. So what ends up happening is you have variation in a population, let's say of bacteria. So the peach ones are the ones that are resistant to this antibiotic. The blue ones are the ones that can be killed by it. So you apply an antibiotic. It kills off all of the ones that are not resistant. But those couple that are resistant survive. You've just done them a favor by getting rid of all of their competition, so they can reproduce very quickly. They reproduce, and pretty soon you now have a population of these guys that is resistant to bacteria, and there's plenty of them. Not just that, but we also learned that bacteria can transfer genes between each other. And so you might have that resistance gene end up on a plasmid. That plasmid gets transferred to another bacterium, and so they're able to spread this even faster. And when that happens in medicine, then you have to find a different antibiotic or a different treatment. And until you do, people are more likely to die. This has not just happened with antibiotics, it also happens with herbicides. So if you spray weed killer on your yard, or you use that weed and seed stuff you can get at Home Depot, there's a chance that the things you're trying to kill, like dandelions or the non-pretty types of grass plants, can become resistant to that stuff. Same thing with pesticides. So roaches, any other sort of insect become resistant to the kinds of poisons that we spray on them. So for example, very soon I'm going to have to get a new can of roach spray because my can is pretty old and there might be some resistance in the population to it. And I don't want to figure that out when I'm trying to spray the crap out of one of those flying ones. So we've affected the evolution of other organisms. Now, let's get to the math. So when you're calculating allele frequency, I think all of this looks a lot scarier than it actually is. I think all the variables and subscripts freak y'all out. So, to calculate the frequency of allele, you need to count up how many copies of that allele there are in a population. Then you divide it by the total number of copies of all the alleles in a population. You also have to keep in mind that for populations like humans, we have 2 alleles per person. So the total number of copies of an allele in the population will be 2 times the number of people in the population, or organisms in the population. You also have to keep in mind that if anyone is homozygous for that trait, they're going to have 2 copies. If they're heterozygous for that trait, they're going to have 1 copy. So if you have 2 alleles, a dominant and a recessive, you have 3 possible genotypes. You can be homozygous dominant, homozygous recessive, or you can be heterozygous. Those are your only possibilities. That's it. So you can calculate the frequency of the dominant allele, and you can calculate the frequency of the recessive allele. They're represented by different variables. So the way I think about it is when you're writing a genotype, like the heterozygous one, do you put the dominant allele first or the recessive allele? You always put the dominant allele first. Well, if you're looking at P and Q, put them in alphabetical order. What are they in alphabetical order? PQ. Which means P goes with the dominant, Q goes with the recessive. That's how I remember it. It's important because, for example, the FRQ that we did yesterday was talking about, it had the Hardy-Weinberg equation, and it was saying, what do each of these variables represent? And you had to know that P was dominant and Q was recessive. Otherwise, you would have missed points. This thing right here, I would just ignore it. It's a little more complicated than we want to get into, especially since you have on average like a minute a question. But if you really want to go into it, this is just saying you're going to take 2 and multiply it by the number of homozygous dominants you have, because they have 2 copies of your allele. And then you're going to just add in the number of heterozygotes you have, because they have 1 copy of your allele. You're going to do the same thing for the recessives. 2 times the homozygous plus 1 times the heterozygous, because that will count up how many alleles you have in the population. And you're going to divide it all by 2n, where n is the number of individuals. All right. We don't have time to do any practice problems today because we still have a bunch of evolution to go. But if you want to, we can always do another session where we do biomath. And in fact, there's going to be an AP Bio Prep session at Townview on April 28th, and they will also have a session that's biomath. All right, so this all relates to the Hardy-Weinberg theorem. If a population is at Hardy-Weinberg equilibrium, we know a number of things. There's random mating, meaning there's no sexual preference, no selection. So it's not like, oh no, I need to mate with the peacock with the biggest feathers. There's none of that. You assume there's an infinite population size. When you get smaller population sizes, the math doesn't work as well. You assume there's no mutations. You want the number of traits you have to be constant. So not only is there no mutation, there's no gene flow. No genes coming in, no genes going out. And there's also no selection of genotypes, meaning there's no natural selection happening. There's no selective pressure. So everyone has the same chance of reproducing. What this allows you to do is to calculate some genotype frequencies if you're given some data. In reality, this doesn't really exist in real life. And if it ever does, it's for a brief period of time. But this is a good model that allows us to make some comparisons and draw some conclusions about how a population might be changing. So the math. So you have these terms in this equation that most of y'all didn't like. P squared plus 2pq plus q squared equals 1. Well, remember that P represents the dominant allele? Well, if you have two P's, that's like two dominants, which is homozygous dominant. So that's your frequency of homozygous dominant individuals. 2pq, well, your P's and your Q's, you have one of each, one dominant, one recessive. So 2pq is your frequency of the heterozygotes. And kind of the reason why it works like that is if you look at a Punnett square. You have one homozygous dominant. You have one homozygous recessive, but you have two heterozygotes. And you could think of that as being where this 2 comes from. Make sense? And then q squared, you have two recessives, and that is going to be your homozygous recessive. Now, the easy thing is 1, both of these equations are on your formula chart, and 2, both of them equal 1. When you add up the number who have, let's say we're looking at short versus tall like we did in our plants. You're either short or tall as an allele. So that means when you add the percentages that have a short allele and the percentages that have a tall allele together, they have to equal 1 because you have to account for 100% of your population. The numbers you get for this, you would get something like P is 0.5. What that represents is if you turn that into a percent, that would mean 50% of your alleles are dominant. If P squared was 0.36, if you're given that information, you can use it to solve for P. How? Take the square root of both sides. So that means 60% of your alleles are dominant, 36% of your population is homozygous dominant. And using that, you can then figure out Q, and you can figure out 2pq and q squared. So once you have one piece of information, you can use it to figure out all the rest. Because I will say, usually you'll see at least one question about Hardy-Weinberg on the test, whether it's a regular multiple choice or whether it's one of the numerical gridded ones. So that's an easy point to get if you get comfortable with the math. So when you don't meet the conditions for Hardy-Weinberg equilibrium and you have a change in allele frequencies, and that happens on just a generation time scale, we call that microevolution. It's not enough to cause the evolution of different species. It is enough, though, if it happens for many, many generations and enough differences accumulate, it is enough to cause speciation death. So let's talk about some mechanisms of selection. So this is what I was talking about with the peacocks. There is sexual selection. It's non-random mating where members of a species show preferences for certain phenotypes. So the biggest examples tend to be birds. Male birds are always really showy. Either they produce these beautiful songs or they have these beautiful feathers. And the reason is the female birds are very picky. And if you don't have the right combination of traits, or you don't have the right song, or you don't build the best looking nest, yeah, you're not reproducing that year. So that's where you get some of these traits that look like they wouldn't be an advantage any other way. Honestly, I don't know how peacocks have avoided getting eaten by tigers all this time. They live in the same place. And I think with that kind of tail, you wouldn't be able to run. I can imagine a tiger just putting his paw on the tail like, you ain't going anywhere. Hello, lunch. But these traits are non-adaptive. They don't necessarily give you an environmental advantage, but they give you a reproductive advantage. So there are 3 different types of selection. And so what this is this is showing the frequency of having a trait, and this is showing like a measurement of the trait. You could do height, you could do the weight of babies, any sort of quantitative measurement of your trait. So you see a relationship in the fitness, and in so what you see is the fitness of these guys for stabilizing selection is usually greatest in the average section of the trait. There's usually some negative selection for the edges. So for example, before modern medicine, the birth weight of babies. If you were on this side, you were too big and you risked causing your mother's death and your own. If you were on this side, you were too small and you were underweight, probably couldn't hold enough body heat to keep warm, and you ran the risk of dying. So only those babies with an average weight would be the ones who would have the best chance of surviving. And so that stabilized the birth weights of humans, so it was in a narrower range. And then you can have something like directional selection. And so you'll have more fitness. Let's see, the highest fitness is over here. That's who's most fit, so that's who's going to reproduce. So let's say this was height of plants. And let's say that it became an advantage to be shorter. What would happen is the ones who were short would reproduce more, the ones who were tall would reproduce less. And after several generations, you'd see a shift in the height, the average height of the plants. So over time, the plants would be shorter and shorter and shorter. That's directional selection because it pushes the graph showing the phenotypes in one direction or another. Then you have disruptive selection where the average trait is not good. So that would be like, okay, it's beneficial if you have a really thin beak or a really thick beak. A thin beak, you can poke in the ground and get worms. A thick beak, you can crack nuts, but if you have a medium beak, you can't do either. And so those are the individuals with the lower fitness, and so they start to make up less and less of the population. They don't survive. They don't pass on their genes, etc. All right, let's talk speciation. So a species is a group of organisms that can interbreed. That is the really common biological definition we use your freshman year. Speciation is when you have reproductive isolation happen where 2 groups are no longer able to interbreed. We have a couple of different definitions of species, but I'm not going to go into them. Let's talk divergent evolution. This is what leads to speciation, where, depending on the environment you're in, if you're in a different environment than another group, you will start developing these differences in your physical or genetic traits that will cause you to be better adapted. So for example, monkeys, primates, our hands are better adapted to being able to climb trees, so hold on to a branch, pick fruit. And so our ancestral group that diverged into primates, the ones who were most successful were the ones who had grasping hands. Whereas the ancestors of whales, the ones who were most successful were the ones who had those flipper-like hands with no separation between their fingers so they could swim better. And so ultimately, when all of those differences start accumulating, you end up with reproductive isolation. The two groups are no longer able to interbreed. They're not part of the same species. Not that we thought primates and whales were part of the same species, but so this is what I was saying. What I was talking about was the biological species concept, where it's like, oh, if they can reproduce, then they're part of the same species. But then you have like asexual reproduction, and so you can say, oh, well, we'll go on like where they are on the tree of life. We'll look at their evolutionary relatedness by like, say, comparing gene sequences. Or you can, if there's some weird stuff that happens with species, you can go with how they look different, but that doesn't work for like birds because the males tend to look different than the females. So there are a number of different ways. So, just because you have differences accumulate between two populations doesn't mean that you have completely separate species. For example, humans. Like 60,000 years ago, a group of humans moved to Australia. And until probably like the 1800s, there was limited interbreeding with the rest of humanity. But they are still part of the same species. They are still capable of interbreeding. And there was probably still some gene flow. There were probably some people like getting in a boat, coming over from Papua New Guinea, coming in from Indonesia, and those people in Indonesia exchanged genes with other humans. And, eventually everybody was exchanging genes. So, in order to separate into two different species, two lineages, you need to have species that are no longer able to reproduce. I think I already went over all this, and we're running low on time. So there are a couple of different types of speciation. Allopatric is when there's a physical barrier. So for example, you had, during an era, the Pliocene era, you had this large area which was actually a continuous lake. And you had species of fish living there. While after you had the glaciers expand and then the retreat of the glaciers, these were separated into different lakes. So the the fish that were in two different lakes that were not interconnected eventually accumulated enough differences to be reproductively isolated, and that ended up causing a diversification in fish species there. You can also have, this is related to like adaptive radiation, you can have a member of a species that can cross a barrier and start a new population, like the finches did in the Galapagos, and then they can adapt to their local environments. Sympatric speciation happens without physical isolation. So usually there's something that isolates them, either their habitats are slightly different. So you could have different species of bird that only stay in, like some of them only stay high up in the trees, some of them may stay only on the ground, and so if they never physically interact, they never reproduce. Or you could have polyploidy. So for example, some plants can develop polyploidy where they have more than 2 copies of each chromosome. Instead of being diploid, they're triploid or tetraploid, and they're no longer able to reproduce with the original population. This has actually happened with one species of frog. I don't remember which is which, but one of those is a diploid frog and the other is tetraploid. They're very closely related species, but they cannot reproduce with each other. You can have hybrids between 2 species, but generally they're going to have some sort of problem. Either they're going to have some sort of, they could have a birth defect, or they could have decreased function of some of their organs, they're less fit, or they could be sterile. So depending on the number of chromosomes each of those have, I believe horses and donkeys have different numbers of chromosomes. And while their genes are similar enough that they can interbreed, mules don't have an even number of chromosomes, so meiosis doesn't occur properly, so they can't produce viable sperm or egg cells, which is why mules are sterile.
Then how do they reproduce?
Mules don't reproduce. You only get new mules by reproducing a horse and a donkey. That's why they're not their own separate species. So we have some isolating mechanisms. Prezygotic is before you have a zygote, so that would be a sperm and egg cell, or that would be preventing those two from ever meeting. Postzygotic is usually something's wrong and the hybrid doesn't survive. So a sperm and an egg cell met, but they produced a less than fit hybrid. So prezygotic isolating mechanisms, you can have the mechanical type, just basically the reproductive parts don't fit together. You can have temporal isolation, they reproduce at different times of year. So if organisms only have a very limited window during which they can reproduce, and if this frog species only reproduces in February and this one only reproduces in September, they're never going to be receptive to breeding at the same time. You can have behavioral isolation. So if you were here yesterday, we did the free response question with the fruit flies and their flashing signals. That was an example of behavioral isolation, because if they didn't have the right flashing signal of lights, they didn't recognize it as a member of their species they could breed with. Same thing happens with calls. So for example, frogs or birds make mating calls, and if their mating calls are different enough, they may be physically compatible and capable of having offspring, but they don't recognize each other's mates. Then you can have habitat isolation, so that's what I described, where if you have some organisms living in trees and others that only live on the ground, they're near each other but they don't ever interact. Or gametic isolation, so their sperm and eggs just won't fuse. Postzygotic mechanisms, it's usually zygotes may not develop into actual ready-to-be-born babies. The adults may not have an easy time reproducing, so they may be mostly sterile, or they may be infertile. You can have hybrids existing for a long period of time. You can have these areas where, like, for example, these are two different species of frog. They have this hybrid zone where the two populations overlap, and they are able to interbreed a little, but the hybrids are usually not as fit as the purebred individuals, and so since they can't interbreed very successfully, they're still considered two separate species. And that is it, right at 5:30.